Creatine gluconate is a form you will encounter occasionally on supplement labels, usually with minimal explanation. The name suggests a creatine-glucose combination, which implies a specific theory about how carbohydrates interact with creatine uptake. A theory that happens to be grounded in real biology but implemented incorrectly in the product. The form is creatine bound to gluconic acid, not glucose, but a closely related compound. Gluconic acid is the oxidized form of glucose, produced when the aldehyde group of glucose is oxidized to a carboxyl group. It has a similar molecular structure to glucose and shares some metabolic pathways. The connection to glucose metabolism is chemical and structural, though gluconic acid does not function identically to glucose in the body. The biological rationale draws on well-established research showing that insulin enhances creatine uptake into muscle. Insulin, released in response to dietary carbohydrates, activates the GLUT4 glucose transporter in muscle cells. Through secondary signaling cascades, insulin also upregulates the creatine transporter, SLC6A8, thus increasing the rate of creatine entry into muscle tissue. Studies taking creatine alongside simple carbohydrates have consistently shown faster muscle creatine loading compared to creatine alone. The argument for creatine gluconate is that the gluconate component provides the glucose-like signal that enhances insulin response and creatine uptake, all in a single molecule. This would allow creatine and its uptake-enhancing partner to be delivered in a chemically bound form. The problem is that gluconate is not glucose. It does not produce the same insulin response. The insulin-mediated creatine uptake enhancement in the research literature was produced by actual glucose, typically 50 to 100 grams of simple carbohydrates, representing a substantial glycemic stimulus. Gluconic acid does not replicate that stimulus at the amounts delivered through creatine gluconate supplementation. The creatine content per gram of creatine gluconate is lower than monohydrate due to the molecular weight of the gluconate component adding to the total molecular weight without contributing creatine. Research specifically on creatine gluconate versus monohydrate for muscle loading is extremely limited. No robust head-to-head clinical trial has established that gluconate produces superior muscle creatine accumulation. The correct application of the insulin-creatine interaction insight is to take monohydrate alongside a meal containing carbohydrates. No novel form required. The insulin-creatine uptake interaction that creatine gluconate invokes is well established in the literature and worth understanding correctly so that you can implement it without a novel creatine form. Studies by Green. Colleagues in the 1990s demonstrated that co-consuming 50 to 100 grams of simple carbohydrate alongside creatine monohydrate produced significantly greater muscle creatine loading than creatine alone. Subsequent research by Steenge and colleagues found that protein co-ingestion also enhanced creatine uptake, presumably through protein's insulinotropic effect. The mechanism is that insulin activates the PI3K-AKT signaling pathway, which among other effects increases the activity and membrane expression of the creatine transporter. More active creatine transporter at the muscle cell membrane means faster creatine entry into the cell during the window when blood creatine is elevated. The practical implementation requires a meaningful insulin stimulus, 50 to 100 grams of carbohydrate or an equivalent protein load. This is the equivalent of a substantial carbohydrate-containing meal. Taking creatine alongside breakfast, a post-workout shake containing carbohydrates, or any meal with significant carbohydrate content achieves this. The gluconate component of creatine gluconate cannot replicate this insulin signal. The dose is too small and gluconate is not glucose. Timing of creatine supplementation in relation to meals is therefore more important than creatine form for someone trying to maximize the insulin-mediated uptake enhancement. Taking monohydrate with a carbohydrate-containing meal consistently produces better loading than taking any form of creatine in isolation. This conclusion requires no novel form and adds no cost. Creatine gluconate is an example of a product built on real science. The insulin-creatine interaction is real, but implemented in a way that delivers an insufficient dose of the active mechanism while charging a premium for the novelty. The underlying insight is useful and actionable. the creatine gluconate vehicle for acting on it is not the right tool. The insulin-enhancing approach costs nothing extra and requires no form switching. Just eat a meal with your monohydrate.